Hi, everyone. Welcome back to our seminar series. For this week, we kind of had to move the seminar from 2 p.m. to 11 a.m., but I'm hoping that this is still working for many of you guys, which is great. And today we are very happy to have Dr. Fei Chen from Broad Institute of MIT and Harvard. And he's also assistant professor in the Department of Stem Cell and Regenerative Biology at Harvard. And his doctoral research time yielded a very exciting technology called expansion microscopy, working with Ed Boyden at MIT. And as an independent fellow at Broad, he led a group which bridged microscopy with next-gen sequencing, and which they called a technology named Slide-seq 1 and 2, right? And that in his current group, he continues to pioneer novel molecular and microscopy tools to uniquely illuminate biological pathways and function. And previously, he was an excellent scholar at the California Institute of Technology and graduated with a Bachelor of Science in Electrical Engineering. And he obtained his PhD from MIT. And with this, without further ado, I'd like to welcome Fei.
Thank you. Thank you, Ahmet, for that amazing introduction. I'm very excited to share our work with this group who sounds like it has similar passions as us. So really today I'm going to talk to you about novel tools for spatial genomics. And as I said right before the intro, our lab also developed some novel synthetic biology molecular recording tools that I won't really have time to share, but happy to talk about that offline. I've been liking to start my talk with this video, and this is like every cell in Drosophila developing embryo from shortly after fertilization to when it's like practically walking off the microscope. And I've always been kind of drawn to this video because it showcases kind of the wonder of biology, but also kind of this immense spatial and temporal coordination that exists, right? And in Drosophila, these developmental processes are quite stereotyped, and we can go in time and time again to label different cells and molecules kind of to build up an understanding of how these processes work. But really in many other kind of biological systems, maybe like the mammalian brain or mammalian development or in disease, we don't have the luxury of such stereotype processes. And so what we really need are tools that allow us to look at this sort of spatial temporal diversity in molecules as we go along. So as it goes without mentioning that one recent technological advance that has really revolutionized our study of molecular mechanisms in cells and tissues is single cell transcriptomics, or in general single cell tools. And this is probably an audience that understands that although in single cells you can break a tissue down into its constituent single cell technology, you can break a tissue down into its constituent parts and sample the transcriptomes of each part. And while this is comprehensive and gives you a look at the cell types and states, you have to remember that we have to dissociate the tissue. And in this case, we lose the kind of the spatial and temporal context of where those cells come from. And that spatial and temporal context is really important, obviously, in biology. For example, cells come together and dynamically form structures and have cell-cell interactions. And not only that, they are they self-organize and divide, and they have dynamic migration or division. And all of these single cell approaches, it's kind of a static snapshot of what happens. So we kind of lose that context of space and time. And so in my group, really, we're interested in bringing kind of these single cell genomics to the tissue context, and in particular, being able to spatially profile molecules in the context of structure of tissue or the structure of cells, and also figuring out ways to look at the dynamics of molecular programs within the context of tissues as well. And so on the side of understanding the organization of tissue structures, there's kind of two critical needs that have been kind of motivated by single cell. One is that, in single cell technologies today, they're quite scalable, and it's actually really quite easy to collect these massive single cell data sets with immense amount of molecular heterogeneity about the cell types. For example, this is a couple hundred different cell types from a very small volume of the mouse brain. But as I said, we actually need approaches to allow us to understand the spatial organization and the connectivity or cell-cell interactions between these cell types. So what we need is high-resolution mapping of gene expression patterns and associated cellular programs and cell types into tissues. And the second, I think, goes without saying, is that we need approaches to relate that gene expression to pathology. For example, for a long time, pathologists can go and look at tissues with two simple stains and kind of identify or classify the pathological features. And at the same time, we've done quite a lot of work on single cell or just genomics on pathology. And we kind of understand the molecular programs that are going wrong. But what we would like to be able to know is to assign, the molecular programs to these distinct and heterogeneous pathological structures directly within pathological tissues. For example, here are just some cases of prostate cancer. The other is that, oftentimes we might have some feature like, here is some amyloid beta in an Alzheimer's model of the mouse brain, and or perhaps injury, and you'd like to know kind of the molecular programs in the cell types that surround these features, and in particular also perhaps the cell interactions of the cells which surround these. And so these are kind of the motivating needs for the development of spatial technologies, something that many of you guys are familiar with. So to set out to do this, actually, to address the loss of spatial context when performing single cell sequencing, we've developed a tool called Slide-seq, which enables genome-wide expression profiling in tissues at approximately 10 micron resolution. And this is work that's really been a joint effort between my group and Ahmet Coskun. Our groups are like down the hall from each other, and we started at the Broad around the same time. And the big picture of Slide-seq is it effectively lets you do untargeted single cell sequencing, and at the same time, it lets you know where those cells came from in your tissue. So how do we do this? How does Slide-seq work? In Slide-seq, we deposit individually barcoded 10 micron polystyrene beads in a monolayer on a surface. And the key is that each bead has a PCR, has tens of millions of DNA oligonucleotide sequences, and each one of these sequences has the same structure. It has a PCR handle, a spatial bead barcode, and this is kind of a clonal sequence that's the same for each bead but different across beads, a unique molecular identifier which allows us to count transcripts, and a poly-T tail which captures mRNA. And what we can do is we can basically take these beads and we can spin coat them onto a monolayer onto a surface of a slide. This is a cover slip that this is 40 millimeters in diameter. Each one of these white circles is a 3 millimeter array of beads. And because these beads are deposited randomly, we can then take a microscope and sequence the clonal barcodes for each bead in the array. So I won't go into the details of exactly how we go and do that, but what happens is that over the course of successive imaging rounds, we're kind of getting the XY location and the sequence associated with each bead in the array. Effectively, it's kind of like a zip code for the clonal sequences. And the process of making these arrays can be done up front, and we've done because the beads are quite bright and 10 microns in size, that this sequencing on the microscope is quite scalable. And so, in our lab, we're regularly manufacturing something like 100 arrays in a week. And so one of the key strengths of this approach then is that once the arrays are made, it's very easy to then perform the actual assay of Slide-seq. So you take the array that's already been made and you take fresh frozen tissue and you section it directly onto the array. And then in kind of a simple protocol, you can generate sequencing libraries very similar to single cell sequencing, where you do kind of reverse transcription and then amplification of the cDNA library. And so when you put that sequencing library on an Illumina sequencer, you get data that's very much like single cell sequencing, wherein for each bead barcode, you have a vector where that vector is the counts that bead saw for each gene in the transcriptome. And so if you arrange all of the beads by genes, that's like a gene expression matrix. Now, if you remember, we have the spatial location of each bead barcode. And so we kind of just match them up. And now you have basically an XY location for each vector in your gene expression matrix. And so because you can do this, Slide-seq actually has very similar structure as single cell data. So we can apply similar computational tools such as dimensionality reduction and clustering. This is actually one of our first Slide-seq experiments where we ran Slide-seq on the mouse hippocampus. And we basically, what we've done here in the very left is we've clustered the beads based on just without any spatial data. And we've colored the clusters by the principal cell types in the mouse hippocampus via the macrogenes. And in the middle, actually, all we've done is we've labeled each location with the color in this unsupervised clustering. And you can see that we recapitulate kind of the structure of the hippocampus, including the localization of principal cell types like kind of the dentate gyrus or the CA1 field. And then kind of here's this oligo layer in the white matter. And so the takeaway is that actually, and then this is actually a serially stained DAPI section that shows that we recapitulate the tissue architecture. We've also performed some pretty detailed experiments to measure the diffusion of transcripts in this process, and we find that it's less than the width of the Slide-seq bead. And so we believe that actually the spatial resolution is effectively 10 microns. And so the takeaway is that there's really no microscope, and we've turned the sequencer into a microscope. And now here we're just showing cell types, but you can actually then generate kind of gene plots for each gene in the transcriptome individually from one experiment. And so, collecting this high resolution transcriptome data allows us to have a platform for scalable measuring the spatial transcriptomics. And it allows us to reconstruct the organization of tissues and connect it to molecular cell types. For example, we applied it to describe the cellular architecture of the mouse cerebellum. And because the size of the bead is about single cell, about 1 cell resolution, we can use it to discover new patterns of spatial variation within cell types. We've actually developed some new algorithms that allow us to look for non-random spatial patterning of gene expression within a single cell type. And we use that to discover spatial gene expression domains in Purkinje neurons of the cerebellum. And lastly, I think, as I mentioned, one of the goals is to be able to visualize spatial gene expression changes in pathology. And in our paper, which I won't go into too much detail, we used it to profile the spatial temporal dynamics of traumatic brain injury. And in particular, we can look at differential gene expression patterns, as a function of spatial localization. So, since our publication of the original Slide-seq paper, which was in 2019, what we've been doing is we've been focused on improving the production and sharing of reagents with the community, including testing in a wide variety of tissue types and organisms, including human, mouse, embryos, organoids, and quite a lot of pathological human specimens, most of which are tumors. And secondly, we've been working on actually the spatial capture yield of Slide-seq. And just through some improvements from molecular biology and bead synthesis, we've achieved about a 10-fold improvement in unique molecules captured per bead to about 1,000 UMIs per bead, similar to maybe first-generation droplet-based single-cell approaches. And this is work that, there's no reason why Slide-seq shouldn't be able to capture RNA at a similar level as single-cell approaches because the underlying technology is the same. So we expect there to be an additional maybe factor of 3 or 4 gains to be had. And so this work is recently published along with some open source array production and computational pipelines, both on GitHub and protocols.io. So, these improvements in Slide-seq has really enabled more sophisticated analyses because we're capturing more transcript per bead. Here, just as a vignette, we profiled the developing mouse cortex at embryonic day 15. And so cells actually here, it's actually a typo. Should be 15.5. So cells here develop from the left to the right here. This is actually the cortex, and the ventricular zone is right here. And they become up here they mature to be part of like the kind of a maturely structured cortex. And we actually developed some tools based on pseudo time or latent time approaches. By examining the coherent paths of gene expression similarities, we can identify genes which vary along this developmental manifold. Using this approach, we can actually discover hundreds of genes which vary along this developmental process. And we can actually then use human genetic studies of intellectual disability and autism to look at how these developmental genes are enriched as a function of where they are localized spatially and where they're localized along this kind of pseudo-time manifold. And and we find things like the relative enrichment of intellectual disability genes is earlier in this developmental process than enrichment for autism genes. And we think that these kind of data sets and analyses will be especially kind of useful for understanding the spatial temporal localization of genetic risk variants in CNS tissues in the future. And so far I've shown you kind of some results from unsupervised clustering of data from Slide-seq. But in many cases, we have great existing single-cell RNA-seq and data sets and transcriptomic annotation, as I mentioned at the beginning of the talk. So what we really like to do is we like to be able to assign beads as like the samplings of the transcriptomes that we find on the array to cell types as classified or that's discovered in single cell. And each bead is approximately the size of a single cell, but it turns out that you can't really control whether a cell lands on one bead or across the division of 2 beads. And on average, it's about 1.5 cells per bead in a typical mammalian tissue. And so what we really like to be able to do, and this causes kind of some issues when we just do unsupervised clustering, in that, for example, when 2 cell types are closely adjacent, Bergmann glia and Purkinje cells, where Bergmann glia are kind of, they both exist in this monolayer in this section of the cerebellum. And when we perform unsupervised clustering, we find beads which have like predominant Purkinje signatures or predominant Bergmann markers, but also beads which have mixtures of both. And really, we want to be able to perform cell type aware analyses, for example, of cell-cell interactions or spatial gene expression profiling within cell types. And we don't want to say that these mixtures are one cell type or other, but we want to know the relative compositions, of cell types. And so we've actually, recently developed a supervised learning approach to decompose these mixtures into single cell types. This is a method called robust cell type decomposition. And the idea is pretty simple. It's to estimate the contributions from individual cell types in a single cell reference to each bead on the array, right? And so, for example, if we have a single cell reference of Purkinje neurons and Bergman glia, and maybe also granule cells, which are another cell which co-localizes them with them. I won't go into detail how exactly the model works, but at the end of the day, we want to say what combination of cell types and in what proportion best fit the observed transcriptome data from each bead. And this is really work that's led by quite a brilliant graduate student, Dylan Cable, in collaboration with Raphael Irizarry. And so, to do this, we've actually performed really detailed computational simulations with single cell data sets where we show that using this method, we can go across platforms. So, for example, one interesting thing that we noticed in single cell and Slide-seq and single nuclei data is that there are inherent technology-specific batch effects. And here, what we've actually done is we've used a single nuclei data set of the cerebellum, and we've used this algorithm to try to classify cell types as measured by a single cell data set of cerebellum. And the diagonal represents kind of the ground truth. And so, so in pure cells, we can identify basically using one modality to classify cell types from another modality. But beyond that, what we can do is we can computationally mix cell types from the single nuclei data and then use the single cell reference to ask basically, can we identify which mixtures they are? And so basically that's this doublet classification problem. As we mix the minority cell type, then as it approaches more than 25% mixtures, we can accurately identify which two doublets they belong to. And more importantly, we can then actually accurately decompose these mixtures into the fractional content of the constituent cell types. For example, here when we mix Bergman glia with all other cell types, we can identify the doublets and then predict what proportion they're mixed with high accuracy. And so this allows us to use kind of single cell RNA-seq references on Slide-seq data to assign and decompose cell types. So going back to our Bergman glia and Purkinje cell example, we show that we can accurately classify singlets, and then for the doublets, we can obtain their doublet proportions and then decompose the proportions into the transcriptome of each cell type. And so why is this important? Well, one is that single cell data sets often have much higher resolution in sampling and the ability to subcluster very fine cell types. And we can leverage that and project those subtype classifications into Slide-seq data. For example, here we projected 27 interneuron cell types into the hippocampus. And one cool thing is that you can actually see that the interneurons are quite big, so they cover more than one bead. And these little clusters are actually some self-consistency within the algorithm, showing that beads that are covering one interneuron are all being assigned to the same bead. Here we're showing kind of subclasses of cortical excitatory neurons. The other thing that's exciting is that there's been a lot of work in detecting spatially varying genes in spatial transcriptomic data. And all of these approaches have really been non-cell type aware when looking for these spatially varying genes. And if you think about it, like the principal component of variation in tissue organization is the organization of cell types. So if you look for spatially varying genes in a tissue in a non-cell type aware way, you're going to find basically marker genes of cell types. And that's just saying that cell types are non-uniformly organized. What we really want to know is find out is how does gene expression change within a cell type as a function of the tissue organization. And so using RCTD, we're beginning to be able to do that. For example, we can discover cell types specific spatial variation, for example, here in the CA3 fields, Or we can find gene expression changes as a function of who your neighbors are. This is some examples of astrocytic gene expression, where the expression astrocytes is a function of whether, for example, here, whether they're near excitatory neurons. So these are kind of some, like SLIC6A11, it's a GABA transporter, and it's expressed in astrocytes co-localized with excitatory neurons. And that kind of makes sense because this is a gene that's known to be associated with perisynaptic astrocytes. And this is just one example, but in general, what we want to do is we want to condition on cell types to kind of deconvolve the contributions of cell type and cellular environment on the heterogeneity in cell states. Okay, so so this is wrapping up this first half of my talk. I want to show that we've developed some experimental and computational tools for high resolution and high sensitivity spatial analyses of tissues. We've developed some technologies that allow us to scalably measure the transcriptomes from tissues, and we have both published some ways to democratize array production and also have internally scaled up our ability to make these arrays. I think importantly, I've shown you how we can leverage single cell atlases to assign Slide-seq data to cell types. And I think the next step is really looking at cell type aware spatial analyses. For example, how do you understand differential expression within the context of cell types and tissues? And then how do you understand cell-cell interactions? And with those tools in hand, we're kind of prepared to make some atlases of mammalian tissues, which is what we're doing in the lab right now. We have ongoing work to develop kind of an atlas of the mouse brain that's about 600 Slide-seq arrays, and some developing mouse work. So I've shown you kind of in this first half of the talk our work at looking at the transcriptome at the abstraction layer of cells. And I really do think that like the 10 micron or 5 to 10 micron resolution is quite scalable for looking at transcriptomes, because we have, the cell as the fundamental like organizing unit of tissues. For this next part of the talk, I kind of want to talk about some new work where we've been doing in situ sequencing. And if you kind of think about the spatial capture work that we've been doing, and also many others have been doing, that's kind of like encoding the microscope with DNA. This other work is actually kind of moving the sequencer into the sample. And so, we recently, and actually this has kind of been work that we've been doing for quite a while now, which is, we've been developing an approach to sequence DNA directly within cells and tissues. And this is really a joint effort led by 3 really great graduate students, Andrew, Zach, and Paul. And it's a joint effort between my lab, Ed Boyden's lab, and Jason Bumroster's lab. And it's kind of seeking to address the same question, right? Which is that, in imaging, we can have these really high resolution, high context, spatial context information. But it's hard for us to access sequence information or content, right? And maybe this is kind of like a lot of what spatial omics is about, is like bridging this gap in sequencing. We have quite a lot of content about exact, DNA sequence or maybe the DNA sequence context, but it's usually we lose the spatial context of where this information comes from. And, I don't want to leave out the fact that, in recent years, there's been a lot of work at trying to bridge this particular question, right? And there's been 2 methods that have really taken off in addressing this. One is DNA fish, and then in particular multiplex DNA fish, which allows you to kind of image genomic loci with predefined probes. Now, one thing is that you don't actually get the sequence context because you're actually you're hybridizing against sequences that you've designed. Here, and another set of aspect methods is Hi-C, which uses like spatial proximity as a way to like ligate nearby DNA fragments. And this has really kind of revolutionized our ability to look at bulk genome structure. But again, this is kind of an indirect way to look at genome structure. So, we set out a couple years ago to ask, can we sequence and image DNA in the same cell? And this turned out to be, quite a challenging and heroic problem, which involved us developing quite a number of new approaches. And so, in our approach, we developed this thing called in situ genome sequencing. And how does it work? Well, because you can imagine there's this parallel where you might you take cells, extract DNA, and you do Illumina sequencing and get genomic loci. Here, what we're actually doing is we take cells, we fix the DNA, and we actually perform in situ library construction and amplification and perform sequencing directly within the the cellular nuclei, and then we map those sequences back to the genome. Now, it turns out that that's actually quite a challenge, and we came up with some tricks to be able to make that happen. The first thing is that we needed a way to construct libraries directly inside of fixed cells, and there we took advantage of this enzyme called TN5 transposase, which is often used in library preparations and in ATAC-seq. We use it in this mode where it's not accessibility sensitive because we denature the histones with a little bit of SDS. We tagment the DNA. The transposase basically inserts adapters into the fixed nuclei. And then what we do is we circularize we use the adapters as handles to do kind of this circularization by ligating these dumbbells on. Now you have a genomic insert flanked by some known sequence and some handles. And this insert now is a circular molecule. So if you put in a 529 polymerase, it allows you to amplify this thing by a rolling circle amplicons and such that each one of these amplicons is now like a clonally amplified dot within the nucleus. And that kind of looks like this. And now the key thing is that it's actually a little bit tricky to do in situ sequencing of many bases, right? Illumina has spent quite a lot of effort and time to be able to generate even short 100, like 250 base paired reads is pretty much an impossibility in situ. But we came up with kind of a clever trick that's a little bit inspired by Slide-seq, is that we can put a UMI into a unique molecular identifier into each one of these hairpins, and now we can kind of sequence this unique molecular identifier in situ, and then we can extract all the DNA and perform Illumina sequencing of both the unique molecular identifier as well as the genomic DNA. And now we can get kind of long reads in situ. So that's basically the workflow. So we perform this in situ sequencing of these amplicons. We actually do 18 bases. We recover them. We then put them onto the Illumina sequencer. And in the Illumina sequencer, we can do paired end reads up to probably 500 or 600 bases. And also we sequence the UMI, we match them up, and now we can get a reconstruction of those long reads in situ. Okay, so we performed a couple of different studies with IGS, including some PGP1 fibroblast cell culture and early mouse embryos. For this talk, I'll just talk a little bit about the data that we developed in early mouse embryos. And so we were really interested in early the nuclear architecture and embryogenesis because it's very clear that in this very in this very early stages of embryogenesis, the chromatin structure and architecture is particularly important. And it's actually a little bit hard to access measurements of single embryos with Hi-C because you can isolate single embryos, but it's a little bit tricky to separate out individual cells with those embryos. And it's difficult to actually also access these with microscopy normally, or a full embryo with microscopy normally, because they're about up to 100 microns thick. We developed some basically advancements in imaging and along with IGS to be able to image them fully. Okay, so we actually kind of wanted to answer a couple of questions, which is one, how does the nuclear architecture vary within zygotes and between developmental stages? And then also, what are the relationships within and between individual embryos? And so we actually carried it out on genome structure at 3 distinct stages. The zygotes, late 2-cell embryos, and early 4-cell embryos. And so it turns out that the number of reads that we can get in situ is directly related to the volume of the cell, and it turns out the volume is relatively conserved as this embryo divides in these early stages. And so we get about 8,000 reads for the zygote, 4,000 reads for the zygote, and 2,000 reads for each cell in the 2-cell, and about 1,000 reads per cell for each cell in the 4-cell. But in addition to this, we can actually combine the reads with stains for kind of protein features. In particular, we looked at lamin, the positions of the centromeres, and also the nucleoli. And so we can now reconstruct basically the localization of individual genomic loci. Here they're colored. Sorry, I should have mentioned that here they're colored by chromosome. And this is a single zygote. And then you can measure the localization with respect to nuclear landmarks. Now, as I said, we actually also get sequence information. So we can use the sequence information to assign kind of the haplotype identities from these individual reads. Here you can actually see the parental and the maternal and paternal chromosomes and SNPs labeled from each one in different colors. And it turns out that in this early zygote, they haven't, the 2 pronuclei are separate. So that's a kind of a positive control for the localization of these individual SNPs. And what we can also, this is kind of just a simple visualization of these pronuclei along with the calls of whether these chromosomes are paternal or maternal. And so this is just like a simple connected dot model where each chromosome position is connected to the next, each localization is connected to the next via its linear genomic position. So it's a little bit like a very rough fold of these chromosomes. Now, you can actually also take this data, and this is not something, this is something we're doing, and you can combine it with high-C data to generate like really high resolution models of these chromosome folds. And this is something that is ongoing. So just for the interest of time, I'll just cover a couple of ideas that we can do. Is that we can look at the organization of chromosome territories, and then also kind of what's happening in both the structure and the distribution of these chromosomes throughout development, right? So right after some of the things that you notice here, right after in 2 cells, when the pronuclei are combined, here are the maternal and paternal chromosomes colored, and you notice that they are not mixed, right? They're actually still separated. And also you might notice that there is kind of, if you look at the centromere to telomere positioning, they're all like positioned with the centromeres on one side and the telomeres on another. And so we actually kind of imagined, we looked into this, and what you can actually see is, and this is something that makes sense, is that, after fertilization and after 2-cell and 4-cell division, you actually get gradual mixing of the paternal and maternal genomes. And you can see that just from this haplotype separation. They're very polarized at the beginning, and then they gradually mix, and we can kind of fit a rate of mixing to this. But also you can see there's actually quite a lot of heterogeneity in the degree that these things are mixed. We can also observe that there's this kind of polarization in the localization of the centromeres to telomeres. This is something that others have observed called the rival configuration. We think this is really motivated by the really fast division times in this early embryo stage. And by associating chromosome features or like localizations of reads to the protein features such as lamina, we can look at, for example, the lamina associated domains and the correlation between GC content and the positioning of the chromosomes to the lamina. And for example, like, you know, this is a well-known phenomenon. Well, not really that well-known, but one thing that we definitely recapitulate is the localization for the paternal chromosomes to be highly correlated in GC content to the lamina, while that is not quite the case for maternal and then after the pronuclei could congregate together. So, and I think, you know, a very interesting thing that we were able to do, because we actually get to localize the structures of these chromosomes within individual cells, but also within individual embryos, is we can look at how like the structures of these chromosomes propagate as a function of like lineage or their relationships to each other, right? For example, you know, within one 2-cell embryo, these two cells are sisters, and we can look at, you know, and thus these chromosomes are separated by one mitotic division, right? And so we can kind of look and see, are the chromosome folds or chromosome positionings more similar in these sister cells than between you know, embryos, between cells of different embryos. And this is kind of like a little bit of a different type of epigenetic information, right? And what we find is that the positioning of the chromosomes within these embryos, for example, here in a 4-cell embryo, the sister cells or inferred sister cells have these really high correlations in the positioning of their chromosomes relative to each other. And actually, you actually see a persistence of this memory across cousin cells. And so here, you know, is the correlation of chromosomes like positioning in individual cells within an embryo and between putative sisters. Here is kind of the correlation. It's still positive for cousins, and then this is obviously null. You would expect that to be centered around 0 for inter-embryo chromosome positioning. So we see kind of like this inheritance of chromosome positioning as a kind of form of epigenetic memory. But we also actually find, and this is some preliminary work, that we actually also find some inheritance of chromosomal folds. And so we're following up on that. And so maybe just as a summary, we've developed kind of this approach, and this is the first approach to allow us to directly sequence DNA within the spatial context of cells and tissues. And we came up with you know, some approaches to enable high throughput, like long paired-in reads, and connecting that to in situ so that we can get both the chromosomal alignment, but also perhaps like SNP and, you know, in other types of tissues, translocation information. And what this allows us to do is kind of look at sequence structure relationships within single cells, and here we did it within single embryos, and also what are the structural relationships between cells, in particular between related cells in an embryo. And so, yeah, to just wrap up, you know, I think my group is really interested in developing tools, what I call like contextual genomics, is really like bringing genomic measurements within the context of structure. And again, it'd be great to talk another time about the context of time. So just, you know, I want to thank everyone for listening, and I'm happy to take any questions. But also I want to acknowledge the folks who actually really did the work, in particular, Ahmet Coskun, and I really have had a great time working together on Slide-seq, and it's really originally pioneered by Sam Rodriguez and Bob Stickles, and now several other members have joined the team, including Evan Murray and Dylan Cable. And as I mentioned, Andrew, Paul, and Zach are really the leaders of the in situ genome sequencing project over the past couple of years, and you know, this has also been a joint effort between Jason, Ed, and I. So thank you, everyone.
Thank you, Fei, for this fantastic talk.
Yeah, it's a super impressive thing. So I think already quite a few questions in the chat box, but since I'm one of the hosts, I would like to take advantage of this this role to ask questions first. I think in your in situ genome sequencing, so do you think you can also look at sort of the dynamic process, for example, cell division, mitosis? That would be super interesting. I don't think anyone has seen that details when the cells divide, the sort of the genome structure changes.
Yeah, for sure. I think, you know, we do capture, it would require capturing quite a lot of cells throughout that. It would be like kind of reconstructing like a, you know, a pseudotime, you know, like trying to like position them in almost like a pseudotime way. One thing is that the number of reads that you get just like directly scales with the volume. And obviously when metaphase, like the volume of the chromosomes like is much smaller, right? Because they're condensed. One thing that we're actually working on in the lab is combining IGS with expansion microscopy, because we really think that that would allow you to get orders of magnitudes more reads per cell. So right now we're sampling the genome at about one megabase or a little bit less than 1 megabase, and in the embryos. And we think that, you know, with expansion microscopy, we might be able to get up to like, you know, 10 kilobase or 100 kilobase sample, which would be really valuable there.
Yeah, that's almost like individual genes, right?
Cool. And we have questions on the chat. Would you like to read them, Fei, or should I help you read them?
Oh, okay. Yeah, I'm happy to read them. Forwards, or I will just go backwards from the beginning. So for Slide-seq, could FFPE tissue be used or just fresh tissue? I mean, this is a great question, and I saw Rong had that great preprint on FFPE tissue, and so I think that it is. We haven't been really working on it, but I think that it's certainly probably possible given like the trails that Rong has blazed. So it is I think probably the thing that is important is the loss in yield. You know, even if like even with really good molecular biology improvements, there was probably still a large impact on yield. And maybe Ron, you can speak to that too a little bit.
So actually, I think our approach in the chemistry is a little bit different, but just want to kind of bring to your attention, to the audience attention, is the Professor Joachim Lamberg actually uploaded about archive paper half a year ago. That's the same chemistry, basically.
Yeah. Oh, sorry. Maybe I'm confused. Maybe I meant Joachim. But yeah, sorry. but yeah, I think they saw a huge decrease in yield. Still, nonetheless, I think it's like a tenfold decrease in yield. So that's something.
Yeah, in our case, about the same thing, about 5 to 10-fold decrease in capture yield or detection yield. Yeah, but the short answer is that works. Yeah, but still some room to improve.
And then Xiaoji Chu says, how about triplet detections and more? Yes, actually that's built into the algorithm. We run our RCTD at doublet mode, but we've actually applied it to Visium data, and you know, you can choose all cell types or triplet or quadruplet mode, depending on what your expectation for number of cells per spot is. We consider that Slide-seq has on average less than 2 cells per spot, so we almost always run Slide-seq in doublet mode. And RNA velocity and pseudotime methods don't explicitly consider spatial information. Do you have thoughts on how to improve those analyses? Yeah, I think this is a great question. We did a little bit of work there on in our paper on how to integrate pseudotime with spatial analysis by just by constructing like a spatially smoothed pseudotime vector, like where you're now considering both the spatial dimensions and the temporal dimension. That worked well because there was one dimension of variation in space, or really two dimensions of variation in space that was continuous. Now, probably more generalized extensions would need to require like thinking about, you know, do you expect cells which are related in pseudotime or state to be related, like close, spatially proximal? Do you expect there to be like sources and sinks? And I think that's a really interesting, like basically research question. And how would you compare disease versus control groups with spatial transcriptomics, taking into account differences in morphology and slicing of the tissue? This is a great question as well. I would say that the case control group analysis in general remains a challenging question for even single cell due to things like batch effects, sampling. We are obviously like things which are really powered with spatial analyses and also in single cells within sample comparisons. For example, you can compare, you know, if you have representations of healthy versus, you know, diseased in the same tissue. And I think working, you know, we are kind of trying to work on the case controls, but it's kind of a challenging question, and I'd be happy to discuss that offline if you're interested.
Yeah, I am, because I'm actually working at the same time with the same issue. So I have a data set of mouse 8 versus 8, and it's kind of challenging as well.
Yeah, it's because it's inherently let's discuss this offline, because you know I can explain why it's inherently a challenging question.
I would be interested.
Chen Wang says, in cases where cell types are not discrete, i.e. cloud of heterogeneous populations, can RCTD be able to deconvolute the cellular composition? This is an interesting question, and you know, I think the approach that I would really think about it is that instead of doing cell types in that case, you might do cellular programs, in which case it would still work. And what by program, I mean like you can do NMF decomposition on your cell states first, and then you can project it with RCTD. But that's a great question. Okay. How much time do we have?
I think that we don't have exact time limit as long as you are available, Fei.
Oh, okay. Because there's a lot more questions, and I'm just reading down to the. I might skip down a little bit so that are the reads on IGS preferentially on open chromatin or evenly distributed on the genome? In our, and this might be related to a previous question, whether you can do a TAC-seq. This is correct. Right now it's evenly distributed because you know, we do a similar treatment that many people do for whole genome sequencing, which is lightly treat the sample with SDS after it's fixed, and that removes the chromatin accessibility. If you don't do that, you can get chromatin accessibility measurements. And similar from Chen Wang, again, can IGS principle be expanded to the transcriptome? I would say yes, and we separately are on I'm on a different manuscript with Ed, where we apply some of those principles in the transcriptome. That recently was published. And okay, so I think this is a really interesting question by Kun Q again. what do you think is the nature of the memory of genome structure, and how many generations are required to erase the memory? So I think, so there's been like some work, a lot of work from a while back where in cell culture, they showed that there was this memory across one mitotic division. And there's been a couple of different theories, none of which have been definitively proven. One is just like how the chromosomes line up in across the metaphase plate could contribute to the single cell division memory. Now, it's a little bit hard to test that hypothesis. And I think that could be partially it. Another feature could be that the cell morphology, like the nucleus like shape, might be like relatively, like the constraint might be preserved across division, and that kind of sets some boundary conditions on where the chromosomes can be. And how many generations are required? I think it's like kind of a decay. We observed it for 2 generations, and probably it exists for 2 to 3 generations. And most sequence spatial transcriptomics just measure 2D space. Do you have comments on how we can move forward to 3D or 4D, including time, on single cell transcriptomics? Yeah, I think that on the 3D side, there's probably a decent number of approaches. Like imaging-based methods are inherently a little bit 3D. And, for example, we did 100 microns for these embryos. Spatial capture approaches, you can obviously do alternate sections. Now, I think some work needs to be done for a computational framework for both aligning and also being able to do analyses across sections. We're trying to do some of that stuff in our labs, but it's an active area of research. And then including time, I mean, that's a totally different question, and I think it would be interesting to think about another time. Okay, I think I got through most of the questions. If people are really interested in the questions I missed, they can feel free to email me.
Yeah.
Yes, thank you. I think
yeah. Also, with regards to your Slide-seq, so now this is really like a 10 micron-ish. So in the past couple of weeks, we have seen kind of people really kind of pushing the boundary.
Oh yeah,
the limit.
sure.
So what's your opinion?
On bioRxiv?
Yeah.
My opinion is that if you look at all those papers, they are still aggregating the data, because obviously you need to, I think that like these gene expression measurements, you need to consider it in the context of single cells. And it could be possible that algorithms like RCTD or these other cell type assignment algorithms will be better as you increase the resolution. But I don't anticipate that you need like 1 micron resolution, for example. I think that there's this distinct trade-off between how much information is in a given pixel and that's just a fundamental limit. There's only like so many RNAs in a given area, right? There's a distinct trade-off between that and your ability to like obtain useful information. So if you're going to be downsampling it later, you should really only be taking it to the point where it helps you assign like aggregate or like assign cell types or like segment the into useful features.
Okay, sounds like probably going to be a sweet spot somewhere in between.
Yeah, I think so. I don't know. I'm curious about your thoughts. We should talk. We should talk about that offline, actually.
Yeah, happy to.
Yeah.
Sounds great. And I think I had one quick question about TN5, Fei. We worked with Howard Cheng before. For imaging purposes, we observed some cell heterogeneity, right? It may work better in
Yeah.
some cells compared to other. So have you had that issue as well?
We've actually worked on that a lot. For attack, you definitely see that, and that's one of the reasons we didn't do chromatin accessibility in our paper.
Oh,
It becomes much better with whole genome, and that's because if you look in the subway of this figure, we found that like if you incubate with heme 5 much longer, things became much more heterogeneous, and that just doesn't work well for attack, because if you incubate it for much longer, you start losing like your chromatin accessibility peaks.
Mm.
Like your signal to noise on actually the chromatin accessibility measurement. Yeah. So yeah, that's a great question.
Great,
So
thank you.
in this case, can you look at other type of kind of epigenetic modification, like a DNA methylation? Yeah, I think no one has looked at that yet, right?
Yeah, exactly. I think that's certainly possible. There's some promising like enzymatic conversion approaches that it really probably depends on how well the enzymatic conversion approaches work. Okay, thank you.
Right, with this, I think let's thank to Fei again. I think it's clapping time. I'll do this digital one.
Yeah, we are doing a real one.
And I appreciate this talk. And before we conclude today, I think we'd like to also announce that next week we will have Dr. Brent Stockwell from Columbia talking about imaging the spatial distribution of metabolites, lipids, and cell types in human and mouse liver, a complementary talk to today's fantastic presentation. And with
so
this,
next
guys,
week back to 2 p.m. Just a reminder everyone, 2 p.m.
Yeah,
Yes.
I apologize for that.
Well, I think, yeah. So when you do really good science, right, this is...
Thank you guys for having me. I really enjoyed talking to you guys.
Of course. Thank you so much, Fei. I appreciate it. And have a nice day. Yeah. See you guys next
Thank you all.
Thank you.
Have a good weekend.